So, protein sorting. All proteins originate on ribosomes in the cytosol, except for some proteins that are made in the mitochondria. And most proteins remain in the cytosol. However, other proteins need to move to organelles, and the way the cell regulates this is through the use of sorting signals on the proteins. This idea of using sorting signals on proteins is what I call a common theme. That is, the cell uses this approach in a number of situations. The sorting signal consists of amino acid sequences that tell the cell where a protein should go in the cell, that is, which organelle or membrane the protein should be associated with. And the sorting signal will even tell the cell whether or not the protein should be secreted. You can think of the sorting signal as being like a postcode on a letter, except it's on a protein. And there are some very well documented postcodes for proteins. For example, this sequence says that the protein, once made in the cytosol, should be imported into the nucleus. This sequence says that the protein should be imported into the peroxisomes. And this sequence is for proteins that should be in the ER, but have somehow ended up elsewhere in the cell. If the machinery of the cell encounters any of these sorting signals, then the cell knows where to send the protein. These signal sequences can be divided into 4 groups. N-terminal signals, which tell the cell to take a protein to a target membrane. Internal stop transfer sequences. These tell the cell to stop threading a protein across a membrane. Internal second signal sequences. These are similar to N-terminal signal sequences and tell the cell to restart threading a protein across a membrane. And C-terminal sequences. These typically tell the cell to take a protein back to where it should be. And combinations of these signal sequences allow the proteins to be targeted and inserted correctly into different organelles and different membranes. Now, I'm not expecting you to remember any of the previously mentioned amino acid sequence postcodes. You just need to know that they exist. However, if you do want to remember one, then the one most people seem to remember is SKL. Serine, lysine, leucine, on the C-terminal of the protein, which directs the proteins to the peroxisome. In the cell, there are 3 types of protein transport. Gated, this occurs between the cytosol and the nucleus using the nuclear pore complexes. Transmembrane, this is a type of transport found between the cytosol and the mitochondria, peroxisomes and the ER. Vesicular, this occurs between the ER and the Golgi, within the Golgi stack, and post-Golgi between the endosomal systems, lysosomes, peroxisomes and the plasma membrane. Okay, so that was a quick overview of protein transport, and we have looked at some common ideas, problems and themes.